let's continue with part 2 of cells, looking at the movement through the cell membranes. Now, back to the organelle cell membrane. The cell membrane, also called the plasma membrane. The job is for the plasma membrane is to separate the outside from the inside and also to regulate what goes through. Now, what goes through is regulated by size and charge and concentration gradient in terms of where's more of it found. So there's pretty much 2 groups of transport. Passive transport, which doesn't require energy and goes from high to low concentration. And then there's active transport, and active transport requires energy. So let's look in your textbook right here. So passive transport, let me get my line because I like to draw lines. Passive transport, passive transport goes across. You've got 2 categories, passive and active. So passive involves movement from high concentration to low, and it uses energy, expends energy. So the examples here are diffusion, osmosis, which is the movement of water, filtration, which is the movement with pressure, usually gravity, but sometimes blood pressure in the kidney, and facilitated diffusion, which is diffusion using specialized proteins. So let's start by looking at diffusion.
Are left alone, they will naturally move about randomly. This means that when we have a group of particles, the particles will tend to spread out. And if given enough time, they will end up evenly distributed. We call this process diffusion. And the definition that you need to know for your exams is that diffusion is a spreading out of particles from an area of higher concentration to an area of lower concentration. Importantly, the movement of particles is completely random. The particles don't choose to go to areas of lower concentration. Over enough time, the particles just end up spreading out due to chance.
So I want you to realize here, first off, that diffusion, random movement of molecules, then you also have it from a concentration gradient. And it will continue, if it can, until it reaches equilibrium. And in equilibrium, that's when you have equal amounts of substance or solute in the solvent. So everything is spread out equally. Okay? And this is important. You've got movement down a concentration gradient, and it just goes with the energy of the system and the energy in the molecules. Good. Second type of transport is called osmosis. Now, osmosis is the movement of water based on the concentration gradient of non-water particles. So here's an example of, you've got your albumin, and albumin is a very large protein. So albumin will not move because it is too large to go through the selectively permeable membrane. So you've got 5% albumin on this side, which means you have 95% water. Okay. Sorry, I'm left-handed and I'm using a right-handed mouse, so we're doing okay, but we're not great. Okay, it's 95% water. Then on this side, you have 10% albumin, so that means you have 90% water. Now, water will move from places of higher concentration to places of lower concentration. So water will move from this side of the beaker to that side of the beaker. Oops, I didn't mean to. So the water's moving from this way to this way. Okay? And it will move until it's spread out. Notice that with osmosis, you're talking about the movement of movement of water based on the concentration of non-water particles. And so this will move until you have you have it equal. Whoops. And when it is equal, you have equilibrium. Just a second here. Erase. Take that off. Move this down. There we go. So it will continue, if it can, until you've reached equilibrium. This is called a dynamic equilibrium because you have movement until it's working. Now, in this situation, it might stop earlier because of the pressure and the size differences. But we'll see. It will move in theory until the concentrations of protein are equal. This is very important in water, or in water movement in cells, because if you have the water concentration in your blood at a non-normal situation, you end up with your blood cells changing. Here's an animation. Let me bring out a video for you. So in this video, we are going to look at blood cells. So here's, let's turn off the really cool music. With the blood, normally the concentration of salt in an isotonic solution is 0.9% sodium chloride. So at that concentration, the salt concentration inside the cells is 0.9. The concentration in the blood fluid is 0.9. So you end up with an isotonic solution, and blood does not, the volume of the blood does not change the volume of the cell. So let's look at some blood cells. So if you were to start at 100 magnification, which is a little more powerful than the rate, well, a light microscope, you'd see blood like this, and then you can change the magnification. This is sort of the high power of a light microscope that you'd get in your high school or in the science lab. What you will notice is the shape. These are beautiful, what are called Bi-Concave Discs. And the reason why they're biconcave, they kind of look like donuts, but there's not a hole in the middle. If you were to take a cross section, you would see that they're thicker on the outside, thinner in the middle, and then again thicker on the outside. This allows for greater flexibility as the cells go through tiny capillaries. And it also has this really large surface area for the transfer of gases in and out, because the more efficient your surface area, the more efficient your diffusion. So it's thin in the middle, but it's not empty in the middle. Good. So that is at 400. So let's go a little more. This is 600. Here we are now at 1,000. And they're clumping a bit. We want to get away from the clumped area. So let's get away from the clumped area. Okay. This is a beautiful example of the cells. So here are the red blood cells, and this one in the middle floating around is a white blood cell. White blood cells have nuclei in them and stuff inside them so that you can see them. The red blood cell, one of the last steps in its formation is to remove its nuclei. So as a result, the red blood cells are very good at acting like boats, but they can't really do as much on a cellular level. The white blood cells are amazing. They can go around, they can search out and destroy pathogens. Fascinating. So here's what the normal red blood cell looks like in an isotonic solution. Let's look at what happens when we speed it up, and then we're going to change the solution outside. So what they did here was add a little more. So they're washing, it's called washing the blood cells, which is basically putting a solution over top of them. Okay, now we can see a change in the shape of the red blood cells. So what we've done here is you added a hypertonic. Hyper means more, and it's in terms of the solute. So there's more salt added. So instead of 0.9, we're at 10%. So that's a huge magnification. And what you'll see is the cells are starting to shrivel, which the name for that is crenate And when the cells shrivel, that is because water is leaving the cells. So if we look at it now, so if the solution around the blood is 10%, that means it is 90% water. And we know from inside the cell, when it was isotonic, it was 0.9% inside, which means it's 99, well, about 99. It's 98. No, it'll be 99.1. Oops, let's go. I can't erase without erasing everything. So it's 99%. Okay, so it's 99% inside. A little bad math there. 99.1 if we want to get technical. Okay, so the concentration of water is greater inside the cell. The concentration of solute or non-water particle is greater outside. So because water is moving from a place where there's lots to a place where there's little, in order to balance it out, the water will leave the cell. As the water leaves the cell is shrinking or crenating. And that is what happens in a hypertonic solution. This is what would happen if you had a lot of salt, such as you're drinking salt water. If you're drinking salt water, it's damaging to your blood cells because all that salt water gets in, it becomes hypertonic once it gets into your blood, and the water will leave the cells. Okay? Hypertonic cells shrink up. Good. Now, when the cells are this shrunk, they don't have the ability to flex as well. So they can damage more. Okay, let's jump ahead now. And now we are looking at adding distilled water. Distilled water is hypotonic because the cells have less. Hypo means there's less water or more non-water particles. So hypo means less. So outside there is less non-water particles. And since this is pure water, that's about 100%. So outside now is 100% water. And inside the cell is 99% water. So the water is going to move from the solution into the cell. And as it moves into the cell, those cells are not very strong, and you end up with cells busting. So let's watch for that. You need to actually watch a specific cell because they disappear. They become larger, more spherical. When they become larger and more spherical, they lose their ability to bend. And now you can see you have a bunch of broken cells. And broken cells are not good because broken cells will not conduct or keep the hemoglobin in and conduct oxygen. So a reminder here that the way I remember it is in hypo, your concentration of non-water particles is low, and that results in your cells to grow. Now, cells don't really grow, they just enlarge and overinflate, but enlarge doesn't rhyme, okay? So hypotonic solution is a solution where you have a low concentration of non-water particles or solute, and it will cause it to grow. So this is very important in terms of keeping your cells healthy, but it's also a way to consider how to pull water in and out of your tissues by adding a protein into, or usually a sugar solution or a protein solution. Anyway, a way of looking at it, okay? So when we look at it, the second type of passive diffusion is osmosis. And then we talk about osmolarity, which has to do with the movement of water based on concentration. The third type of passive transport is filtration. Now, filtration occurs when there is some sort of push. And in this situation, in blood pressure, the push has to do with blood pressure. You may think of filter, if you filter something such as coffee or dirt, when you filter something, you use gravity to pull it out. In filtration, you've got the push of the blood pressure pushing things out. So that is filtration. Good. The next type of diffusion is facilitated diffusion. And in facilitated diffusion, you have special channel proteins. And so here you can tell this is a membrane because here you have your little phospholipid bilayer. Here's your little phosphos and your lipids down here. And here is a protein that goes all the way through.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier protein would then transport the molecules out of the cell.
The first type of transport was passive transport. The second one is active transport. Now, in active transport, you are going up the concentration gradient, which means you're going from lesser to greater, such as swimming upstream, and that takes energy, and the energy is in the form of ATP.
The second category of cell transport is active transport. This is when particles move from an area of lower concentration to an area of higher concentration. When particles move against the concentration gradient, energy is required, often to allow protein pumps to assist in particle movement. Why would the cell need to move particles from a low to high concentration and expend energy to do it? An important example is seen in your heart muscle cells. In order for your heart to beat, there are certain molecules that have to move from an area of low concentration to an area of high concentration for those cardiac muscle cells to work. So the main things to remember are: passive transport happens automatically with no energy required, while active transport needs energy for it to occur.
The example of transport or active transport in your textbook is the sodium-potassium ATPase pump. So when we talk about nerves and muscles, we talk about how there is more green guys, potassium inside, and more red guys, sodium outside. And it is pumped this way against the concentration gradient. And this pumping will then set up this concentration gradient that when you open the cells with a nervous impulse, you have reaction occurring. So that is one type of active transport. And you can tell this is membrane channels because here's our little membrane or phospholipid bilayers. The second type of active transport is actually engulfing things or making vesicles of the whole membrane. So in something called endocytosis, the cells come around and will completely engulf it. Here's an example of endocytosis, a type called phagocytosis, because the phago means eating. So here we have a white blood cell and here we have some bacteria. And what the white blood cell does is just engulf the bacteria. And you can see it puts it into its own little vesicle. And then there is some specialized vesicles called lysosomes. And the lysosomes will then fuse with the vesicles with the bacteria. And they digest on their own without it ever being in the cell cytoplasm. So this way you do not destroy the cell itself. So that is endocytosis because it's coming in, and it's phagocytosis because it is big cells.
Large particles, however, pass into the cell by endocytosis. This is an active process. It needs ATP. In this process, the plasma membrane surrounds the particle, such as bacteria or a food morsel, and forms a vesicle that pinches off from the membrane. The cell does this in two different ways. One way is called phagocytosis. In this method of cell eating, large particles are surrounded when the cell extends the plasma membrane and engulfs the particle. The result is a membrane-bound package of material. Fluids that the cell wants to take in enter by a process called pinocytosis. In this type of endocytosis, the cell membrane sinks in, the material sinks inward, and the membrane pinches off. The cell uses energy for this movement. In both types of endocytosis, the cell ends up with a membrane-bound vesicle. The material in the vesicle still has to pass through the membrane in order to be used by the cell. In this case, the lysosome joins with the vesicle and digestive enzymes break down the material to particles small enough to pass through the membrane and into the cytosol. A specialized endocytosis called receptor-mediated endocytosis also occurs in cells. In this process, molecules of a particular substance, such as cholesterol, bind to receptors in the plasma membrane. The vesicle is formed as the membrane sinks in. Then a lysosome joins the vesicle and the digestive enzymes break down the substance, releasing it from the receptors. The substance diffuses into the cytoplasm, the lysosome reforms, and the vesicle, with now empty receptors, becomes part of the plasma membrane again. This allows for the cell to reuse or recycle the receptors. A function of some cells is to manufacture or synthesize materials for export. In many cases, this material is made up of large molecules like proteins that cannot pass through a plasma membrane. The process of exocytosis enables the cell to pass this material out of the cell.
I really like that diagram or that video because it shows how exocytosis will take the vesicle, and these are the proteins that have been made, if you remember back to your organelles, so they were coded for in the nucleus, made with the ribosomes when the messenger RNA came out of the nucleus with the ribosomes, matched up with the transfer RNA, and then that then went into the endoplasmic reticulum. In the endoplasmic reticulum, the proteins were then folded and shaped. Then they go to the Golgi, and in the Golgi they fuse with lipids, and then the Golgi will send off these vesicles, which then fuse with the membrane, and these specialized proteins are released. Now that was a big thing on transport. The textbook has a really good summary on it at the end. And I'm going to go and start another video, part 3, for mitosis, meiosis, and cell transcription and translation.